Hi guys, welcome back to the PhD Prep Lab channel. I hope you all doing well. So now your ICMR examination is very near and the dates can be out anytime now. So the time will be very limited. And since the CSIR exam is done, now you can fully focus on this exam, and this is our like best chance for a rapid revision. So we are starting a rapid revision series, especially for Part B for ICMR examination, as this section is going to be common for all the students, irrespective of your Part C. So today we'll start with the rapid revision of genetics and genomics, as you can see. And this topic is very important for not only your ICMR JRF examination, but even for other examinations like your DBT, GATE, etc. So before we start the video, I want to quickly remind you that we have already put up a video on biosafety and bioethics topics. There is a complete like one short video on biosafety bioethics and uh, available on our channel, and then we have another video where we practiced questions completely based on biosafety and bioethics. So we have two videos on biosafety bioethics, one short video and then a question practice video. So make sure that you have not missed out on those two videos, and we leave the link for them in the description box. So make sure that you check those two videos out. And now let's get started with this video. Let's practice for part B, and let's start with genetics and genomics. So for this video particularly, we'll focus on Mendelian genetics, microbial genetics, and the genetic disorders. So these three are the most important uh, core of the genetics and genomics. We'll mainly focus on these three, and other uh, uh, areas of genetics and genomics are definitely also covered, but these three are like the most important, where questions are definitely asked every year. So we make sure that you focus on these topics more. So now we'll start with the video. Okay, let's start with the first topic. Okay, now we all know what genetics is. It is the study that deals with heredity and variation. Now, what is heredity? It is the transmission of the traits that you get from your parents, that the parents transfer to the offsprings. That is called heredity. And variation is the similarities and the dissimilarities or the differences that you see in a population of a particular species. Like, for example, human population. We all don't look the same. We are in so many we look different, we have different hair, different eyes, different hair, everything is so much different. That is the variation that we see amongst the human population. Okay, and now when we talk about genetic material, so we have DNA, which is the genetic material in us, and DNA is a double helix structure. It is made up of nucleotides that are joined together. Now, which nucleotides? Quickly revise: adenine, thymine, guanine, and cytosine. That is your purine and pyrimidines. Okay, and when we talk about RNA, so now thymine is replaced by uracil, so RNA will have adenine, uracil, guanine, and cytosine. Now, the genetic material will all control all the biological processes. It will maintain, develop, and also monitor the reproduction of the organisms. Now we have a quick timeline here how genetics inheritance study it all came into play. So it all began in the 1850s, where Mendel came in. He worked on peas, Pisum sativum. He proposed the laws of the genetics. So this was all done in the beginning of 1866. Then Fleming came in and he observed mitosis. Now came the 1900s, where Sutton published the chromosome theory. Now, in 1905, sex chromosomes were identified. Then, 1910, Morgan came in. He proposed the linkage theory about the gene linkages, etc. Then, in 1928, Griffin proposed the transformation experiments. Now, in where he the experiment that, if you remember, was done on the mice, introduced with R strain and S strain. So make sure you go and do a quick revision on that. Now, then we have on 1944, Shargaf. First of all, McLeod, McCarthy, and Avery, they came in and they proposed that DNA is the genetic material. Then Shargaf determined the nucleotide ratio. You know that ratio, right? Shargaf's rule, ATGC, etc. Then came in the 1950s, where Rosalind Franklin, he gave out the X-ray image of DNA molecule, and in 1953, Watson and Crick published the DNA double helix work. Now, 1958, the DNA polymerase was used to build DNA molecule, and in 1966, mRNA and amino acid codons were identified. Now, in 1970, the discovery of restriction enzymes were done. Then, in 1972, first recombinant DNA were produced by Berg and Boer. Now came 1985, where PCR was developed by Gary Mullis, and in 1997, Dolly sheep was cloned, and in 2000s, Human Genome Project was completed. So this is the timeline. So this timeline is mainly to show you who, like the important names of the scientists like Mendel, McCarthy, who gave the transformation principle experiment, like Griffith. Okay, so the scientist names are very very important, so make sure that you remember these here. Maybe not so much, but definitely the scientist name and what they have done, so that's really important. Now we have the Mendelian principle. So these are the very core laws of genetics. Broadly, they are the base of the genetics. Okay, these laws determine the base of the genetics. So these laws were obviously given by Mendel. That's why they are called Mendelian laws. So the very first law is the law of dominance. So in a heterozygote, okay, where we have two different kinds of alleles. So one is the dominant allele and the other is the recessive allele. And the dominant allele will always mask the expression of the recessive allele. So when you have, example, a allele for height, you have a gene for height and you have two alleles, capital T for tall height and small t for small height. So capital T will always, so in presence of capital T and small t, so capital T, the dominant height will always show. So this is the law of dominance. Now we have the law of segregation. So where alleles, they separate during gamete formation. So you have alleles, right? Alleles are basically the two different types of genes, right? Like you have one gene for height. Now you have two alleles for the alleles could be tall for tall height and for another allele for dwarf height. So these are the alleles, and these alleles, the capital T small t or small t or capital T, so they separate during your gamete formation and which occurs during anaphase 1. So your law of dominance and your law of segregation, they were based on monohybrid cross. This also you have to remember, this could be asked. And now you have independent assortment. This was performed and explained using dihybrid cross, and this states that alleles of different genes, they segregate independently, and this happens during metaphase 1. Now alleles of different genes means, suppose you have a gene for eye color and you have a gene for height. So these are two different genes. Now these genes will also have two alleles. Okay, you could have a allele for recessive allele for blue eye and a dominant allele for brown eye. Now similarly, you have height alleles, capital T and small t, tall and dwarf height, right? So these two are different genes. So they separate independently. It's not the blue eye gene allele will not go where the height alleles are going. Like they are segregated independently of each other. These traits, they don't pass together, any trait, okay, unless they are linked. So this is the what the independent assortment states about, that when you have different genes, alleles of those different genes, could be any genes, they separate and segregate independently during metaphase 1. So you have the key ratios here, remember it, and also remember, uh, make sure that you know what they are asking, whether they are asking genotypic ratio or they are asking phenotypic ratio. So for a monohybrid cross, you have the genotypic ratio 1:2:1 and the phenotypic ratio 3:1. Now, genotype for the test cross progeny ratios are also given. It's always going to be 1:1 or 1:1:1:1. Now, for dihybrid cross, you have this ratio genotypic 1:2:1 2:4 2:1 2:1. Okay, so remember, and the 9:3:3:1 is a very common ratio, but you have to remember this is the phenotypic ratio. They can confuse you, they can ask you genotypic ratio, you might, and they will give you this 9.9:3:3:1, but you have to make sure that you're able to differentiate between genotypic and phenotypic ratios properly. Now, moving ahead. Now, these are the concepts of genes. Okay, now you have alleles. Now, this, I told you, this is the alternative form of a gene. Now, you have multiple alleles. So, when you have more than two alleles in a population, for example, your ABO blood group type, you have you have alleles for A blood type, B blood type, and O blood type, and AB as well, right? So this is multiple allelism. But any individual in a human in an individual, there will be only two copies, okay, of an allele out of whatever number are present in total. Now you have two formulas for the genotype. So this is n plus 1 by 2. Make sure you remember. Sometimes they ask. Now, what is a pseudoallele? Now, pseudo means false, right? So they are the genes that are functionally allelic, but they are structurally non-allelic. Allelic forms, they are those that are present for the same, they represent the same character, like allelic forms could be your allele for A type, B type, O type, or AB type. Now, non-allelic forms, they do not represent the same character. One could represent a character for height, one could represent for hair texture, one could be for your hair color. Now you have some other important points as well. Now you have complementation test. Okay, this is a very important concept about complementation. So in complementation, what happens is we determine in complementation test, we determine whether two mutations are affecting the same phenotype, are in the same gene or in different genes. Okay, same gene or in different gene, that the two mutations are present. Which what are they affecting? The same gene. The phenotype that they are affecting is in the same gene or in the different gene. So from this, we can also kind of know that whether the genes are likely on the same chromosome or on the different chromosome. So that is very important. So the questions are very trickily formed. They will give you a complementation test very in 1-2 lines, and then they will ask you to identify whether these this gene is present on the same or the different chromosome. So, and you have to identify using the results of the complementation test. So when see how we interpreted the results is when two recessive mutants are crossed, okay, recessive mutants, and the F1 offspring shows the normal phenotype, like the wild type phenotype. The mutations, it means that they are complementing each other. Okay, so from recessive mutants you get when a wild type offspring, it means that they complement each other, and when they complement each other, so complement each other means they're getting the wild type. So when they complement each other, when they get the wild type phenotype, so it means that each mutation is present in a different gene. So this is a very important um point. So they will just simply give you this question in this format only, that two recessive mutations are crossed and F1 offspring shows the normal wild type phenotype. So now what do you know about this? Are the mutation present on different genes or on the same genes? So you have to say that mutation are present on the different genes since they are complementing each other. And when there is no complementation and when you cross the mutants, so you will not get a wild type phenotype, you will still get a mutant phenotype. So this means complementation has not occurred and both the mutations are on the same gene. Okay. Now, what do you mean by wild type allele? Like the most prevalent allele in the population, like the normal allele. okay, the common allele. And the mutant allele is which that produces the phenotype that is not typical to the population. Now you have reciprocal cross. So reciprocal means reverse, right? So this is just the reverse cross, what you take for male and female. You just reverse it in the F2 generation. Okay. Now you have another two types of crosses. First, in the broader sense, you have a back cross. Okay. So in this back cross, you have two types of crosses. First is out cross, and then you have test cross. So cross is basically in which we cross the F1 generation with a parent phenotype. Similar to the parent, okay? So when the parent phenotype is dominant, when we cross the F1 generation with the dominant parent, it is called outcross. And when we cross the F1 generation with the recessive parent, it is called the test cross. Now there is a important note, which is that you have to always check when you always want to check the genotypic confirmation. When you want a genotypic confirmation, we always perform a test cross. We don't perform an out cross. Okay, as in that the dominant parent is used, dominant genotype is used, so the dominant genotype will only show up. So basically, test cross is performed to confirm the genotype of the F1 generation, whether it is heterozygote or whether it is homozygote. Okay, now there are some gene interactions. You should study about these gene interactions in detail. Okay, so since this is a rapid revision, so we'll only look at the important aspect, which is the ratio, which is very, very important. They directly ask the ratio for dominant epistasis, complementary gene, like that. Okay, so here the epistatic ratios we'll study. So the interaction type is dominant epistasis, so the ratio is 12 is to 3 is to 1. The recessive epistasis ratio is 9 is to 3 is to 4. The complementary gene ratio is 9 is to 7. The duplicate gene ratio is 15 is to 1. The ratios are very, very important. Now you have other concepts like your incomplete dominance. So here you get typically three phenotypes. Okay, the heterozygote also shows a different phenotype. So the ratio here is 1 is to 2 is to 1. Now you have codominance. So both alleles are expressed, like your AB blood group. Okay, A is also dominant, B is also dominant, but AB is also, when they are present together, they show AB blood group. Now you have penetrance. It is the percentage of the genotype showing the phenotype. Okay, so you suppose you have a dominant gene, and that in that the penetrance is of the gene is 60%. So it means that 60% population will show the expression of the dominant gene. Now you have expressivity. It is the degree of expression, how severely the gene is affecting. Okay, the severity of the expression basically. Now you have pleiotropy. Simply means one gene with multiple traits, like your phenylketonuria. Now you have phenocopy, where the environment affects the genotype. Okay, this the example is thalidomide. One more important point about phenocopy is that this phenocopy is non-inheritable, since it is induced because of the environment. It is the environmentally induced phenotype, so it resembles the mutant phenotype, but it is non-inheritable. Okay. Now we have mapping, and in this we have linkage mapping versus we have physical mapping. So you have to very importantly remember this. Okay, so you have this table where we have differentiated for ease of remembering and revision. Okay, so your linkage mapping was basically given by Morgan and Strutevant. So this measures the frequency of recombination, and it correlates with distance. And the map distance is given in centimorgan, that is C small and M capital. Okay, the formula is recombinants divided by total multiplied by 100. And 1% recombination, when it is given, it means 1 map unit or 1 centimorgan. And maximum recombination frequency is always 50%, which occurs between the unlinked alleles. Okay, unlinked alleles means they are not linked, they are present at a distance on the chromosome. Now you have 2-point cross, which is the distance between A and B, and the 3-point cross, which determines the order. The double crossovers are the rarest. So the order means when you have to identify which alleles are present in which order. Is it ABC or ACB, like that? Okay. Now we have physical mapping. So in physical mapping, somatic cell hybridization is there. So you fuse the human and the mouse cell. Okay, so it is using the somatic cells. It is done using the somatic cells. So of human and the mouse cells, here we have taken the examples, and the human chromosome is lost completely. Okay, so basically it is to study the gene location, gene expression, and the chromosome function. So it uh, correlates the gene product with retained chromosome. Now you have certain under physical mapping, only you have to remember that you get molecular markers. Okay, sometimes questions could be very tricky, like you have so and so molecular markers given. Now you have to identify whether it is used for linkage mapping or physical mapping. Okay, like that. So you have to remember that under physical mapping, only molecular markers, they come into play. So you have plenty. You have RFLP, RAPD, ALF, AFLP, SSR. So apart from these, a lot of other markers are also present, but these are the most important ones. Okay, so your RFLP is a non-PCR marker, you have to remember it. And RFLP and SSR is the codominant marker. Okay, so it means that it can identify the heterozygous condition, heterozygous alleles. But your RAPD and RAFLP, so these are the dominant markers, only can identify the homozygote dominant condition of the uh, alleles. Now we come to microbial genetics. So under this we have 3 types. So like basically, what are these? Conjugation, transformation, transduction. What is all this? So this is basically the 3 main mechanisms of horizontal gene transfer in bacteria. Okay, so first you have conjugation. So this is done using this is basically the direct transfer of DNA from one bacterium to the other using cell to cell contact. Okay, in this you have a F plasmid. This is the F positive. This is sorry, the F plasmid, wherein you have an F plus cell and an F minus. So F plus is the donor and recipient is the F minus cell, and it involves the conjugation involves a sex formation of a sex pilus through this is the basically the physical mode of transfer. Okay, the pilus forms between the two bacteria, and the DNA is usually transferred. Okay, like that. So here you can see the formation, how it is done, right? And the F minus plasmid is integrated into the genome. The transfer of chromosomal genes happens, and now we have your transformation. So it is the naked DNA uptake. So basically, the DNA is taken up from the surrounding of the bacterium. There is no physical contact. Okay, so this is transformation. This was discovered by Griffith. You remember their transforming principle, right? So this was come from there. Okay, it requires competence and it is sensitive to DNAs. So you have co-transformation here, which is the frequency used to map close genes. So the Griffith experiment was done in Streptococcus pneumoniae. Also remember this point, and he obviously gave the transforming principle. So there is basically no cell-to-cell contact. This is what you have to remember. Only cell-to-cell contact is found in conjugation. So the in transformation, the DNA can come from a dead bacteria also. So basically, the environment when it has DNA, the other bacteria takes up that DNA. So now you have transduction, which is virus mediated. The first 3 points of these three modes of transfer of gene are very, very important: cell to cell, naked DNA uptake, and virus mediated. Okay, coming back to transduction, this is the virus mediated gene transfer. Okay, it is special, like you have generalized and specialized. So in specialized you have lytic cycle, and sorry, in specialized you have lysogenic cycle, and in generalized you have lytic cycle. So basically, this is mediated by a bacteriophage, and the DNA is protected inside phage coat. This is highly efficient gene transfer transduction. Okay, so in generalized gene transduction, as I told you, any bacterial gene can be transferred using the lytic cycle, and in specialized transaction, only specific genes will be transferred using the lysogenic transfer. Okay, lysogenic cycle. Now, another very, very important point. Here you have your pedigree analysis. Definitely one question minimum you will get from pedigree analysis. So you have autosomal dominant types, you have autosomal recessive type, you have X-linked recessive and X-linked dominant. So obviously, always you have to start from the proband. Okay, whenever the pedigree is given, you always have to start from the proband, and then you have to go up and up each generation to identify what is happening, which form is it? Is it first you'll try to identify between autosomal and X-linked, then you will identify between a dominant and a recessive. Okay, then you can narrow it down. So in autosomal dominance, there will be no gene skipping of the generation. Okay, the affected child, if there is a child affected, it will definitely have the parents, both of the parents affected. One example is Huntington disease. Also remember the examples. Now you have autosomal recessive. Autosomal recessive disorders, this skips the generation, and mostly it is seen in consanguineous marriage. It increases the risk there. And consanguineous marriage is marriage between the relatives. Then you have the examples of cystic fibrosis. Now you have X-linked recessive disorder. This is the crisscross mechanism. The males are mostly affected. So we have only mentioned the most important points. Okay, so males are more affected in X-linked disorders, and this is usually transferred from mother to the son. Example is hemophilia. Now you have X-linked dominant, where if you have an affected father, all the daughters will be affected. This is a very important point. So these small points that are given are going to be very, very useful for you to identify the type of disorder correctly. Now you have extra chromosomal inheritance, which is the maternal inheritance. It is uniparental from mother to the offsprings. So this is seen in mitochondrial and chloroplast DNA. So these DNA are passed via egg of the X cytoplasm, and the sperm contribute no cytoplasm. So father has no contribution in the extra chromosomal transfer here. So Mirabilis jalapa, the 4 o'clock plant leaf variegation, is determined by the female parent branch. Now you have petite in yeast. This is the respiratory deficient mutant type. So this is also an example of your extra chromosomal disorder. So in extra chromosomal disorder, remember one main point: the phenotype, like whatever the genotype of the mother is, that will only affect the child, not the father. So we have different types of mutation. We have transition, transversion, frame shift, and nonsense mutation. Definitely one question you will get from here. Okay, so you have transition mutation where purine to purine changes there. Adenine will change into guanine, or pyrimidine to pyrimidine. Cytosine could change to thymine, or thymine to cytosine. So these are the single base pair changes. Okay, purine to purine, pyrimidine to pyrimidine. Now you have transversion where purine to pyrimidine changes there. So from adenine, it could go to cytosine. Okay, so there is mispairing. Adenine to thymine will not happen because that is the correct pair. Adenine to cytosine can happen, or cytosine can add it, cytosine to adenine. Then guanine to thymine change can occur and thymine to guanine. Okay, so purine to pyrimidine change, that is transversion. They will give you a code and you have to identify which mutation is happening. Is it transition, transversion, frame shift, or nonsense? Okay, now in frame shift, um, basically you see a shift in the frame of the codons. Okay, so they alter the reading frame. So if there is a change in say the one of the codons, so the entire frame, like the all of the codons are affected from the point of mutation. Okay, so I will give you an example. So suppose you have a code given, okay, and there could be insertion or deletion of one or two nucleotides only, okay, which can alter the triplet reading frame of a gene. If there are three nucleotides added or removed, it will not disrupt the entire frame. Okay, then likewise you have to remember in exam. Okay, so there is a nonsense mutation also, which basically where the stop codon gets created. So wherever the nonsense mutation occurs, there is a stop codon that has been created due to insertion or deletion of a codon, and there only the like the truncation of the protein will occur. So now you have recombination, wherein you have first homologous recombination that occurs between during meiosis of in the prophase 1, and also seen in the repair of double crossover, double stranded break of double stranded break repair. You also see the homologous recombination. So there occurs a formation of the holiday model. Okay, so holiday model is the formation of the heteroduplex. Okay, so under homologous recombination, some other points that I want to mention. So basically, homologous recombination is what? It is recombination only. It is a genetic process. So in this, DNA strands are exchanged between two DNA molecules that share extensive sequence similarity, ensuring the DNA repair and obviously the DNA diversity occurs during meiosis prophase 1. So you will in this, like I told you, the holiday model will be formed, and in E. coli, Rec A is responsible for this type of strand invasion, and Rec B, C, D is the helicase or the nuclease. RUV, ABC is the branch migration or resolution point in E. coli. So these are the points. Okay, make sure that you go through the theory, because if you don't know the basic theory concept, you will not understand this slide. So now you have gene conversion as well, which is the non-reciprocal transfer of the information. Now about transposons. So these are also called the jumping genes. So these are the mobile genetic elements. It was given by Barbara McClintock. So just try to remember the name of the scientist as well. Sometimes it could be asked. We don't want to lose marks unnecessarily, right? So discovered by Barbara McClintock in maize plant. Okay, so she basically showed that genes can jump and alter the gene expression. So there is a big theory behind this. So make sure that you go through it, the theory part, the base part. So basically divided into two classes, class 1 and the class 2. The class 1 is the retro type of transposomes and the class 2 is the DNA based. Okay. So the DNA transposons, they so first let's do actually the class 1, okay, retro transposomes. So the mechanism here, importantly remember the mechanism of the class 1 type of transposomes are the copy and paste, okay, and the examples are line, sines, ALU, TY elements. And the class 2, like the DNA transposomes, the mechanism that they follow is the cut and paste mechanism. So copy and paste and cut and paste is very, very important. And in cut and paste, the examples are the ACDC, the F elements of Drosophila. Now you have polygenic inheritance. So what is polygenic inheritance? Poly means multiple. So here multiple genes are residing at different loci contribute to a single trait. So one single trait affected by multiple genes. Okay. So remember this. Each loci, the gene location basically is referred to as a quantitative trait loci. So these points are very, very important. And each gene is typically having a small effect on the trait. So multiple genes, one single trait affected. So each gene will have small, small contributions, small effects on the trait. And the cumulative effect of these traits that result in the observed phenotype.
So now the example that is very common in polygenic inheritance that we see here is in the kernel color in the wheat. And if there are 3 genes involved, the phenotypic ratio that you will be getting is this that you can see on the screen, and you will get 64 different types of genotypes. And if there are 4 genes involved, then the phenotypic ratio that you will get is this different one, and you will get 256 types of genotypes. So you should remember this, how many types of genotypes you will get if 3 or 4 genes are involved. Now concordance. So it basically refers to the probability that a pair of individuals will both possess a specific characteristics or a phenotypic trait given that one of them has it. So basically concordance is studied for the twin studies. So the gold standard for concordance is the twin studies. So here we see that the same phenotype is occurring or not in both the members of a twin pair. Okay, it refers to the study of the presence of the same trait or disease in both individuals of a pair. The pair here refers to the twins. So if both the twins show the trait, the diseases, so it is concordant in nature. And if only one of the twin shows the trait, so it is discordant in nature. So it helps to distinguish between the genetic versus the environmental effects of a particular trait or a disease in specifically twins. So if the concordance is higher, so it means there is a stronger genetic contribution of the gene or the disease in the twins. So in generally monozygotic twins, they show 100% concordance generally, but in dizygotic, it's 50% or even lower expected concordance percentage. So here is the genetic disorders chart. It is very important. You will definitely get 1 or 2 questions from this. Remember the examples. It is very, very important. Okay. So for autosomal dominant, you have Huntington's disease. And then for autosomal recessive, you have 4 examples that you need to remember. That is cystic fibrosis, sickle cell anemia, phenylketonuria, Tay-Sachs disease. Under non-disjunction, you have Down syndrome, trisomy of 21, and Turner syndrome, Klinefelter syndrome as well. For deletion, you have Cry-Duchat syndrome, that is the deletion in chromosome number 5. And X-linked recessive disease is the hemophilia A that you need to remember, and the red-green color blindness. So all the examples are very important under the respective type of genetic disorders. Okay. So make sure that you remember this and not forget it in the last moment. Definitely 2 questions at least you will get from this portion. So now moving on. This was it from the part B revision of genetics and genomics. I hope that you liked the video. I hope that you find it useful, and this is the beginning of the series of the rapid revision. We will cover every chapter that is there, every unit that is there for the part B of ICMR JRF examination. So it will be very useful for you guys. And make sure that you follow us and subscribe to our channel so that you don't miss out on these videos. And also we have already uploaded the biosafety bioethics one-shot video and the MCQ practice of it. So go check that out also. The link is in the description box, and you can join our Telegram group where you can get instant updates for your 2 different groups are there, for JRF notes and for internship job opportunities. So the QR is given for both the groups. Make sure you join both of them. Important information is shared there. So we are keep building a community for specifically your examinations, internships and notes. So if you like the video, give it a thumbs up, and I wish all the best to all the participants. Make sure that you stick to our series and do the rapid revision of part B chapters units with us and ace your ICMR JRF examination. So all the best, and follow us and subscribe to our channel. Thank you for watching.